Gracias, Íñigo, por la presentación y a ustedes por estar aquí presentes. La charla que les voy a dar es prácticamente la misma que di en la defensa, lo que les voy a ahorrar es la lectura de las conclusiones finales y espero que la disfrutéis. La verdad que por logísticas no me dio tiempo antes de la defensa de hacer un ensayo aquí en la casa y me sentía en deuda de que vieran el producto final, ¿no?, de todos estos años de trabajo. Es una tesis que ha sido dirigida por Pedro Jordano y Alfredo Valido y que lleva por título Opportunistic Pollination by Birds and Lizards in the Canary Islands, y la voy a dar en inglés, que es tal y como la di el día de la defensa. Espero que no sea un problema. Bueno, empezamos, ¿no? The key question around which we designed this thesis project is the following. What determines the reproductive outcomes of plant-pollinator interactions? And all of us know that there are many factors that have this influence on plant reproductive success, but there are two important aspects that are common to any plant-animal mutualisms that are pollinators' effectiveness and the ecological context in which this interaction occurs. With respect to the pollinators' effectiveness, it's largely dependent or influenced by life history traits of pollinators. All of us know that there are a huge diversity of pollinators all around the world, and they differ in the cognitive systems, physiological systems, learning abilities, foraging behaviors, etc. And this huge variation determines an ample variation at the end in their performances and in their delayed consequences on plant reproductive success. Among these pollinators, we can find both vertebrates and invertebrates. And with invertebrates, it's possible to distinguish between those that are specialist nectar feeders, that are vertebrates that mainly or largely depend on nectar for food, from those that are opportunistic nectar-feeding vertebrates that exploit only occasionally this floral resource for their diet, and for their survival. These opportunistic nectar-feeding vertebrates are frequently or particularly frequent on oceanic islands, and this higher frequency has been hypothesized as a product of ecological release. And the logic of ecological release is the following. Oceanic islands has a lower number of species compared to the mainland, and this lower competition of species, along with a lower risk of predation and parasitism, lead vertebrate populations in the islands to increase their abundance by density compensation, and at the end, this higher abundance leads them to a higher intraspecific competition, forced the individuals to exploit novel food resources such as floral resources, nectar or pollen. So they play important roles as mutualists for insular flora. Some examples of these archipelagos are those that you can see in the slide. And a priori, we could expect that due to this lower dependence on nectar, opportunistic vertebrates would be less effective than specialist vertebrates. But first of all, to test this hypothesis, it's necessary to determine what makes really an effective pollinator. Well, pollinator effectiveness is a matter of quantity and quality. Quantitatively, a pollinator is effective when it visits flowers with high frequency, deposits more pollen grains on stigmas, and in this way, there's a higher probability of ovule fertilization. And qualitatively, a pollinator is effective when it deposits and transfers pollen grains to suitable stigmas, so the probability of siring viable seeds is higher. For opportunistic vertebrates, we would expect a lower quantity and quality respect to specialist nectar feeders because they would be less consistent in their floral visitation, and there would be a greater stochasticity in the origin of pollen because they are more dependent to the availability of alternative food resources that maybe are more preferred than floral resources. But apart from their life history traits that are intrinsic to these pollinators, there are also other factors that affect or impact their quality and quantity as pollinators, and this ecological context in which the mutualism is surrounded. Among these factors, we can distinguish the biotic environment, co-flowering plant species, and non-pollinating interacting agents. And in this thesis, we focus on the role of these non-pollinating interacting agents that we term floral antagonists. And these represent a very diverse taxonomical group that can counterbalance the reproductive benefits made by our opportunistic pollinators by direct and indirect effects, directly consuming male and female gametes of plants and indirectly altering the foraging behavior of pollinators. Both aspects that we mentioned before, pollinators' effectiveness and the ecological context, has been widely studied in how they impact plant reproductive success, but the interplay between these both factors has been understudied, especially in the case of opportunistic pollinators. And this is the challenge that we try to meet with this thesis project. To meet this challenge, we chose as our study system a particular groups of vertebrates that are passerine birds and lacertid lizards that are distributed in the Canary Islands. This archipelago belongs to the Macaronesian region that also include other archipelagos like Azores, Madeira, Salvage Islands, and Cape Verde Islands. And this region has an apparent paradox because its flora contains several plant species that are adapted to bird pollination, but especially nectar feeders are absent from this region. So the evolutionary origin of this adaptation remains unclear. And the current situation is that these opportunistic vertebrates are their current floral visitors, and they interact with these plants in relatively high frequency in the field. Despite this high frequency in the field, it wasn't until the 1980s when the first list of potential ornithophilus plant species was proposed in the case of the Canary Islands by the great pollination biologist Stefan Vogel and collaborators. And since then, the number of interactions has increased. So the metrics of interactions has reached up to now 56 different interactions between plant species in the rows, that are 19 species, and 11 species of birds and lizards. But we know that this matrix is also underestimated, and in the future, new interactions will appear. Despite this high frequency of interactions, the point is that their effectiveness of pollinators had not been tested before. And this point, this lack of knowledge, constituted the basic step and the starting point to design our thesis project. So concretely, we posed 4 questions. The first one, this one, are these vertebrates, birds and lizards, really effective pollinators of the Canarian flora? Once we ask this question, then we get inside the pollinator assemblage that the plant species receive to see if the life history traits of birds and lizards determine that they are not functionally equivalent or yes as pollinators, and the absence or presence of the functional equivalence will have important consequences for plant reproduction, as we will see later. And then we ask the last 2 questions, jumping from a pairwise perspective to a more integrative perspective in which we include all the signs and magnitudes of interactions that the plant receives during the flowering season, including not only the mutualisms, but also the antagonists. And we ask concretely how the combination of effects of mutualism and antagonists determine the spatial variation that we find within the plant population in plant reproductive success, and how this variation in space emerge from the overrepresentation of determined interacting animal assemblages. We used as our main response variable throughout the whole thesis, female plant reproductive success measured as fruit and viable seed production. And we chose 2 study systems that were precisely the 2 first plants confirmed as visited by these opportunistic pollinators by Vogel and collaborators, Isoplexis canariensis and Canarina canariensis. Both are mainly distributed in the laurel forest, that is a relictual forest of the tertiary, and both are distributed in the western islands of the Canary Islands, but they reach their highest abundance in the island of Tenerife. So we chose our 6 study populations in this island, concretely in 2 regions that correspond to the 2 oldest geological areas of Tenerife, Teno Massif on the left and Anaga Massif on the right. So starting with the first question, are these vertebrates really effective pollinators of the Canarian flora? we had to start from the very base because there was a huge lack of knowledge of the natural history of this plant species. So first of all, we had to determine their breeding system, if they were or not compatible, the importance of their pollen origin for their reproduction, and we did it by hand pollination experiments. Then we determined the whole spectrum of floral visitors and their visitation rate, and finally, from these different guilds, who were the more effective or the most effective in the fruit and viable seed production, including our opportunistic vertebrates. This last one was made by exclusion experiments. So for the breeding system, we found that both species are self-compatible, they accept their own pollen, but the origin of pollen has a great impact, and the most interesting result was found with the proportion of viable seeds. Here we can see that in both species, those flowers that were pollinated with pollen from other individuals that are in white produced a higher percentage of viable seeds compared to those treatments or those flowers that were pollinated by self-pollen. And curiously, these flowers pollinated with cross-pollen produced similar proportions of viable seeds than those that were opened and without any treatment, and open and potentially visited by our opportunistic vertebrates. Respect to the floral visitors, we found for Isoplexis canariensis that all visitors, floral visitors were opportunistic vertebrates with the potential of being effective pollinators because they contacted the reproductive organs. And we found as the most frequent species a bird, Phylloscopus canariensis, the Canarian chiffchaff, followed by a lizard species, Galotia galoti, and the rest were all bird species, but in a marginal representation. The situation for Candelaria was, by contrast, different because our opportunistic vertebrates occupied lower positions in a quantitative point, and the first positions in floral visitation were occupied by slugs, solitary bees, and introduced rodents that acted mostly as detrimental agents, floral herbivores, and pollen thieves. But despite these differences, our opportunistic vertebrates were, among all these functional groups, the most effective ones because they increased or enhanced the female plant fitness. And the result is shown in this figure. No? For both species and for fruit set and viable seed set, we found that those flowers exposed to our opportunistic vertebrates in white produce significantly more fruits or more viable seeds than those flowers that were exposed to their influence. So for this question, we can conclude that yes, indeed, they are effective pollinators of the Canarian flora, at least for these two plant species. Once we have checked this, we get inside the pollinator assemblage and try to identify how the life history traits of birds and lizards, who are really contrasting organisms, can determine the level of the functional equivalence of the pollinator assemblage that our plant species receives. For this, we focus for each animal group in the two most abundant species that we mentioned before. Phylloscopus canariensis as a representative of the bird functional group, and Galotia galoti as the representative of the lizard functional group. Concretely, we posed the following questions to test this difference and this equivalence. First, do they differ in the quantity and quality component? And secondly, how these differences in quantity and quality determine the level of the functional equivalence for the plant. And in the case of insular plants, it's really important if they are not, or they are functionally equivalent, because the pollinator assemblages of insular floras are smaller, depauperate, and more disharmonic compared to the continental islands. So against any disturbance, the risk of extinction is greater compared to the mainland flora. So returning to the formula that we introduced in the beginning, pollinator effectiveness is defined as the product of the quantity and quality, and the subcomponents that we defined for each one was, for the quantity, the product of the number of visits that the pollinator made to the plant per unit rate, no? In our case, per 30 minutes per plant,
multiplied by the number of flowers that the pollinator brought per plant visit. And within the quality component, we wanted to use surrogates of male fitness and female fitness. As a surrogate of male fitness, we use the proportion of pollen grains removed per single floral visit, and this surrogate was added to the product of the fruit set produced per floral visit and the proportion of viable seeds produced per floral visit. This formula led us to calculate an overall value for each species of pollinator effectiveness and plot both functional groups in the pollination effectiveness landscape. In a few words, the pollinator effectiveness landscape is a bidimensional space defined by all combinations of quantity and quality, and within it, it includes an elevational contours of isoclines of a pollination effectiveness from this corner to the upper right corner. So each isocline defines all these combinations of quantity and quality that renders the same value of effectiveness. For example, 0.5 can be reached by a high quality and a low quantity, or the inverse, a low quality and a high quantity, and in the middle, all possible combinations. In our case, what we found? We found that birds were much more effective pollinators than lizards because they were not aggregated in this landscape. And this large difference in effectiveness was mainly due to the subcomponent of the visitation rate that was much higher in the case of birds compared to the lizards. And we hypothesized that it's due to the metabolic constraints of lizards. No, birds are endothermic, they can visit flowers throughout the day, and indeed that was the situation that we saw in the field. Whereas lizards, their activity is restricted to the hottest hours in the day. But we found another interesting result, and was when we analyzed the pollination effectiveness at the interspecific level, because we found that birds, adult and juveniles of lizards, despite they had an overall similar pollination effectiveness, they differed in the quality component, and specifically the difference was due to the proportion of viable seeds that they produced. So adult lizards produced a higher percentage of viable seeds compared to the juveniles, and we hypothesized that it's a consequence of the pattern movements, so maybe adults are promoting a higher rate of cross-pollination compared to the juveniles, no? So we conclude for the second question that our pollinator assemblage, due to their disparate life modes of birds and lizards, determine that they have, this plant has a lower functional equivalence. And this could be a signal, a critical signal for a greater risk against a drastic reduction in the abundance of the most effective pollinators, or even in the most severe cases, against to their extinction. Okay, when we have analyzed how the life history traits affect their consequences in plant reproduction, we jumped to the more integrative approach, in which we include also the effect of antagonists. And it would, in these last questions, emerge from our experience from the field, because we saw that maybe different plants were highly benefited by pollinators, but despite of this, the reproductive outcomes were completely different. So there was something, another source of variation that was promoting this spatial difference in plant reproductive success. And in this case, this third agent were the floral antagonists. So we asked the following question, no? How the effects of both agents determine the spatial variation that we see in plant reproductive success within the plant population? For this, we defined 6 functional gates within each group. For mutualists, as you already know, we defined the following 3: legitimate birds and legitimate lizards that visit flowers properly because they contacted the flowers without any detrimental action. No? And the third one of facultative bird pollinators that sometimes switch from legitimate to illegitimate behavior or vice versa. This group was really contrasting with the antagonistic group formed by invertebrates that acted as floral herbivores, nectar lacertids, and predispersal seed predators that were smaller in size, that had a limited movement compared to the vertebrates because they mostly interact with the plant in the larvae phase. So in the larvae phase, so it determines a higher interaction intimacy with the plant, and the timing of interaction because, for example, floral herbivores were able to start their damage since the bud stage, and predispersal predators could act at the end of the flowering season before seed dispersal, whereas our pollinators are restricted to act when the flowers are open. So what were our expectations in relation to the spatial structure of the interaction strengths with mutualists and antagonists, considering these life history traits? For the antagonists, since they have limited mobility and a higher interaction with the plant, higher interaction intimacy with the plant, we would expect that they, first of all, couldn't contact it with all plants within the population because they have this limited mobility. And given their higher interaction intimacy with the plant, we would expect that those nearby plants would show similar interaction strengths compared to the rest of clumps within the population. So at the end, we would expect a greater spatial signal, a greater spatial structure in their interaction strength. And the opposite trend for pollinators, because they are highly mobile organisms, they can move easily between these patches of plants. And along with their opportunistic pollinators, as they are able to exploit other alternative food resources, their interaction intimacy is not going to be so strong. So we would expect a lower or absence of a spatial structure in their interaction effects. And the combination of both interaction strength within each individual plant would determine at the end the spatial structure of plant reproductive success, being able to find hotspots and cold spots of plant fitness. So the concrete questions for this chapter were, are these interaction effects of antagonists and mutualists spatially structured or not? If so, how the combination of both the spatial signals or structures determine the spatial structure of plant reproductive success? And within all the 6 functional groups, which of them, if there is the most important in driving this spatial difference in plant reproductive success? For this, we chose to replicate within our study population, and for each plant, we determined their X and Y coordinates, a series of plant traits to control their potential confounding effects, the interaction strength with mutualists and antagonists defined as the product of the frequency of the interaction and the intensity of the interaction, and after the flowering season finished, we determined the female plant reproductive success as the product of the fruit set and viable seed set per fruit. These variables, to see their spatial structure, were analyzed using a spatial pollen pattern analysis, concretely marked correlation functions, and using the summary statistic of Slater's index. And the graphical representation of this analysis is the following that you can see in the screen. This is a spatial autocorrelogram, and what you can see is the different distant classes, and for each distant classes, the Slater index measures the level of a spatial autocorrelation of our variable or combination of variables, including all pair of plants that are separated by this concrete distance. We could find a positive spatial correlation if the value of the Slater index falls outside the simulation envelopes of our null model that expect that the values of the variable are completely randomly distributed in the population, and negatively correlated if the value falls below the simulation envelopes, and no signal at all if they are included within the envelopes. So in the case of the effects of mutualists, we found that there was a strong spatial signal, a positive signal. So plants that were separated up to 3 meters approximately were more similar than expected by chance in their interaction with mutualists. And it was counterintuitive to our expectations because for antagonists, we find also a spatial signal, but a weaker one compared to the mutualists. The most interesting result was that when we analyzed the spatial association of both effects of mutualists and antagonists, we found that there was a spatial repulsion between both. So a plant or a focal plant that had a high interaction effect with mutualists was surrounded by plants with a low interaction effect with antagonists. So in the population, we could find hot and cold spots of plants with high interaction with mutualists and low with mutualists, and inverse. But how these effects, the combination of these effects, determine the spatial structure of plant reproduction? First of all, we had to determine if this plant reproductive success was a spatial structure, and yes, it was also. Curiously, up to positively correlated, and up to 3 meters, similar to mutualists and antagonists. And the question was, which of the two functional groups were the drivers of this spatial structure that we find in plant fitness? And the result was that this signal in plant reproduction didn't reflect the spatial structure of any functional group in isolation, but a combination of both, because plant reproduction was positively correlated with mutualists, but also negatively correlated with antagonists. So we can conclude that plant fitness is the net effect of the counterbalancing effects of both functional groups. This result was also corroborated by our linear regression analysis, in which we found that the interaction strengths of mutualists and antagonists were significant on plant reproduction, but the interaction strength of antagonists was greater, 2.5 times greater than that of mutualists. And when we scale down to the 6 functional guides, we found that the functional guide that had the highest impact on plant reproductive success were floral herbivores. And we hypothesized that they are the most important because they start their impact on plants earlier, the earliest action of all these functional guilds, since flowers are in bud stage. So they create like a template of plants that are, some of them damaged, others not, before the arrival of pollinators. And when pollinators arrive, we assume that pollinators are able to discriminate this damage, and in consequence, they avoid those clumps of plants that are damaged, and it results at the end in hot and cold spots of plant reproductive success in the population. Given that we find the spatial repulsion between mutualists and antagonists in their effects concretely, and that the plant reproductive success is a spatial structure, it led us to the last question of the thesis and try to understand how this spatial structure emerged from the overrepresentation of determined interacting animal assemblages, more than expected by chance. How to detect these overrepresented interacting animal assemblages? With the help of a network theory, concretely with the help of the identification of interaction motifs. Interaction motifs are considered in this network theory as the building blocks of the structure of complex networks, and are subgraphs that are overrepresented, that are more frequent than expected by chance within these networks, and are the ultimate drivers of the design of the whole network interaction. So in our case, we constructed and we built a bipartite network between the set of our plants, the same as the one used in the previous chapter, and our two functional groups, mutualists and antagonists. And for each individual plant, we proposed 3 alternatives in their interaction. consider, for example, interaction with antagonists, no? The first option is that we didn't detect any interaction at all with this animal functional group. The second one, that they indeed had an interaction, but this interaction is weak. Or the last one, that this interaction is strong. And the same three possibilities were proposed for the mutualistic interaction. At the end, we obtained a total of 9 possible interaction typologies that were later summarized in 6 interaction typologies, just to integrate the whole complexity of interacting animal assemblages that we find in the population. And these 6 typologies were arranged along a gradient from a mutualistic gradient, where we can find plants that interact exclusively with mutualists, to the other extreme, where plants can interact exclusively with antagonists. And in the middle, all possible combinations of plants that interact with both groups, but with different alternatives of interaction strengths. From these 6 typologies, as we said, we were interested in detecting which ones were the overrepresented in the network, which were the interaction motifs. But apart from this, we wanted to go a step further and try to discern what were the functional consequences for the individual plant of belonging to one or another interaction typology. Concretely, what were the functional consequences in pollen transfer and in the female plant fitness, no? In the production of viable seeds and fruits. Concretely, with respect to the pollen transfer, what we would expect is that plants that were interacting exclusively with mutualists would be more integrated in the mating network. So they would occupy more central positions in this mating network. So they would act with a higher probability as pollen donors or as pollen sources. And this centrality in the mating network would decrease as we move to the antagonistic extreme. Just consider that this is a graphical representation of our mating network in the population, in which the nodes are the individual plants and the links are the number of events, of mating events, that we find in the population between these 2 plants. And in our case, this number was determined by the number of bird flights that we observed in the field, no? Between each pair of plants. We would consider a central plant within this mating network, those plants that had many connections with the rest of conspecifics, but also the one that had the shortest direct and indirect paths to the rest of the members. So compared to those plants that had a marginal position in this mating network, they would reach faster the pollen grains from the conspecifics, or export fastest their own pollen grains. But apart from our expectation in the centrality, according to this gradient, we also would expect that as we move from the mutualist to the antagonistic extreme, plant fitness would decrease due to the direct and indirect effects of the antagonists. So the concrete 2 questions for this last chapter were the following. From the 6 typologies that we defined, which ones are the motifs? And once detected, what are the functional consequences of these interaction motifs in pollen transfer and in plant reproductive success, no, for the individual plant? Again, we use the same replicate plots that we used in the previous chapter. And for each plant, we determine their interaction typology, the plant centrality within the mating network, and the female fitness. And those were our results. For the first question, the results of our motif analysis, what we found is the following. No? In this graph, we see the 6 interaction typologies, and then in the y-axis, the frequency of each of them. The observed frequencies that we obtain in the field are represented by the field bars, and the expected ones by randomizations of our interaction network are in white bars. And we, concretely, in our study population, we identified 2 interaction motifs from the 6 that were these ones, no? This and this, that had two characteristics in common. First, that are hybrid motifs, that are diversified motifs. That means that the most interacting assemblages were those plants that interacted with both groups, mutualists and antagonists, but also that they were hybrid motifs in the sense that they don't have a similar interaction strength with both groups, but there are 2 alternatives. Or you interact with the plant strongly with mutualists and weakly with antagonists, or the inverse. And this result is in concordance with the previous chapter where we found the spatial repulsion between the mutualistic and antagonistic agents, and that is driving ultimately the spatial structure that we found in our plant population. This gradient from mutualism to the antagonistic gradient was correlated or associated with a decreasing trend in the plant reproductive success as we expected. So as plants are more damaged by antagonists, the reproductive benefits are counterbalanced, and you produced as an individual plant a lower proportion of fruits and a lower proportion of viable seeds. But our expectation wasn't fulfilled with respect to pollen transfer. So we didn't find, as the more central plants, those that were more mutualistic, those that belong to the most mutualistic interaction typologies. Those are the mating networks that we found in the 2 replicate plots. And as you can see, within the central positions, we can find both types in the sense that you can find mutualistic and antagonistic interaction typologies, and even those plants that are completely mutualistic typologies are even occupying marginal positions. So which of the 6 were the most central typologies, concretely? No? This result can be easily seen in this graph, in which we show the relationship between the plant centrality and the female reproductive success. What we obtained is that, okay, if you are a more central plant, you have a greater plant reproductive success, although this relationship is relatively weak. But this increase in plant centrality associated with female reproductive success don't coincide with our gradient in typologies, because we don't see a gradient from orange colors that are the antagonistic typologies to the more blue colors that correspond to the mutualistic typologies. No? What we would expect is precisely that the fully mutualistic interaction typologies would fall in this region. But instead of this, we obtain that plants with these colors that coincide with these ones are the more central ones, and are plants that have a certain level of damage with antagonists. So what does it mean is that in some way, the damage of antagonists at certain levels even favor the integration of the plant within the mating network compared to other plants that are only visited by mutualists. And we hypothesize that it could be because certain levels of damage in the plant don't avoid completely the visitation by birds, but the resources are not sufficiently enough because of the damage by antagonists, so it promotes the movement of the bird or the lizard to other plant within the population, promoting this higher integration of the plant. But if the damage by antagonists is really strong, as this is the case, the plant is completely disconnected from the mating network. It can't contribute with their genotypes for the following generations. So I would like to finish the talk with the main contributions of the 2 blocks that we defined from the beginning. So in relation to the life plant traits, life history traits of pollinators, we conclude that our opportunistic vertebrates are really effective pollinators, but they are not functionally equivalent. And we understand that we need further research in this area because, first of all, we have beginning to describe how is the role of these opportunistic vertebrates in the Canary Islands, but the metrics of interactions is still underestimated. We need more field observations to really complete this matrix of interactions. And apart from this, we only have tested their effectiveness in only 2 out of the 19 plant species that are visited by them. So it's necessary a greater effort to have a proper framework of their importance in the ecology of evolution of the insular populations, not only in the Canary Islands, but in the whole Macaronesian region. And respect to the second block, the antagonistic context, we conclude that it's really important to have an accurate prediction of the plant reproductive success, the inclusion of different types of interactions with the plant, not only the interactions with mutualists, but also with antagonists, but not only taking into account the sign of interaction, but also and explicitly the interaction strength of this interaction, because the results could be completely different, as we have seen in the last chapter. Y me gustaría finalizar ya en mi lengua materna, en español, agradeciendo a todo el equipo humano que está detrás de este trabajo, en especial al grupo de investigación al que pertenezco y en concreto a mis 2 directores, a Pedro Jordano y Alfredo Valido. Nuevamente les doy las gracias por todo el apoyo que me han ofrecido, que ha sido constante, y por todo lo que he aprendido tanto a nivel personal como profesional de ellos. Luego también a los 2 grupos de investigación con los que pude realizar las estancias durante la tesis, que es el grupo de Ana Traveset en el Instituto Mediterráneo, Estudios Avanzados, y el grupo de Steve Johnson en la Universidad de KwaZulu-Natal, en Sudáfrica. Y por último, pues a la gente de mi tierra, en concreto por un lado a las organizaciones públicas y no gubernamentales que facilitaron todo el tema de los permisos, y todos aquellos colegas naturalistas, amigos y muy especialmente a la familia que me echaron un cable tanto con dudas de historia natural como permitiendo y ayudándome en el trabajo de campo. Y a todos ustedes por vuestra atención y vuestra asistencia.